Bismillahirrahmanirrahim. Welcome back, guys. Welcome to another video sessions from Kame biology. In this video lecture we will talk about the virus purifications. Fine. So now what is virus purifications? Virus purification means that let's suppose we have a tubes. Within the tubes we put the sample of the patient's blood sample or sputum sample or any kind of sample. We will put liquid. I'm talking about the liquid sample. Fine. So we will put it into the tubes, and once we put it into our tubes, so what we do, inside these samples, the sample will contain the cell, the sample will contain the viruses, the sample will contain the cell contents, sugar, and all of the mixture the sample will contain. Fine. So within each of the mixture, how we can isolate the active viruses, or how can we isolate the viruses which cause the infection inside the human being. Fine. So how we can do that? All these techniques are called virus purification. Means that how we isolate the active viruses from in this mixture tubes, in this mixture solution, how we can isolate the viruses. Fine. It is a kind of experiment. We always perform this experiment inside the lab. So now so now in this topic we will talk about the virus purification. So now let me write virus purification. Now for virus purification, so we take the sample, blood sample or urine sample for the viral infected patient. Once we take that sample, then we transfer it into a tube. Fine. So now let's suppose we take the sample. Fine. And we transfer it into a tube. This is the first tube. So what we do here, we transfer the sample. Fine. So we transfer the sample here. The sample should be blood or urine. Once we put the sample here, so now this sample will contain, let me draw some diagram. These samples will contain the viruses. This green one represents the viruses. Fine. Now the sample will also contain mitochondria. Let me draw. This blue one represents the mitochondria structure. Fine. It also contains Golgi bodies, Golgi apparatus, and endoplasmic reticulum, all of the cell organelles. And it also contains the cell. Let me draw the cell with a black one. Let me draw. This black one represents a cell, and there are some other cells which are infected by viruses. So within each cells there are viruses are also present, but now these viruses are active viruses. These viruses are mature viruses. When the viruses are present inside the cell, animal cell or plant, obviously these are the human cells because we take the sample from the human. So if the human cells contain the viruses, so these viruses inside the cells, these viruses are immature. One thing, these viruses are the complete viruses, and these viruses are the infected viruses as well. So we just need these viruses. Fine. So what we do after that, we put some detergent. Fine. So we put some detergent, or we put the buffer. So now what the buffer and what the detergent actually do? The buffer, once we loaded the buffer inside each of the tube which contain the viruses, which contain the ribosomes and protein, and which also contain the mitochondria and other cell organelles. Fine. So now within each inside this test tube, so once we loaded the buffer or detergent, so what actually it do, it just rupture the cell. Fine. So now the cell is ruptured. So once the cell is ruptured, what we get the mature viruses or the complete viruses. Now these green ones represent the complete viruses. We get the complete viruses. Fine. So we rupture the cell with the help of detergent or buffer, and then we get a complete viruses. Inside the cell there are also the organelles are released out. For example, endoplasmic reticulum. Let's suppose this one represent the endoplasmic reticulum. We also get the endoplasmic reticulum from each burst of the cell or other cell organelles. Fine. Now this solution is contain a lot of mixture: protein, mitochondria, Golgi bodies, viruses, mature viruses, incomplete viruses, and all of the organelles are present. Fine. But how we can isolate the viruses from each tubes? So for this purpose, what we do, we centrifugate the tube. So we proceed the tube for the centrifugation at 10,000 speed. On that speed we centrifugate it for 1 to 3 hours. Fine. So for 1 to 3 hours at 10,000 speed, on that speed we centrifugate the tubes. Let's suppose C represent the centrifugation means. Fine. So we, after the centrifugation, what happened in this case, let's suppose we centrifugate this at 10,000 speed for 1 to 3 hours, and after the centrifugation then we get the supernatant. After the centrifugation, within the tubes we get the supernatant. What is supernatant actually? Let me explain. These are the viruses present. These greens represent the viruses present. Fine. Inactive or incomplete viruses, and we also get some pellet after some times. We get the pellet. Now this black one represents the pellet. Fine. And this one, as I draw here, this one represents the supernatant. Fine. So we get the supernatant. Now the viruses which are obtained from this supernatant, these viruses are now in inactive form, or we can say these viruses are incomplete viruses, or we can say these viruses are not mature viruses. So for this purpose, what we do, we just take these viruses with the help of pipette. Means that we take out these viruses, inactive or incomplete viruses, with the help of pipette, and we discard it. Fine. So once we discard it, then we resuspend these pellet once again. So after that we discard this and we resuspend the pellet. So what we get, now we get now let's suppose inside these tubes there the supernatant are discarded. So now within these tubes there are the pellet. Now the pellet contain cell content, organelles, and active viruses. Active viruses means complete viruses, or the viruses which cause the infection inside the host cell. We can also say that. Now these viruses actually we need for our further procedure or our further test. Fine. So we need these viruses. So what we get the pellet, and now we resuspend the pellet, and again we get the active viruses. Let's suppose this time these green viruses represent the active viruses, and other cell content are also present: mitochondria or ribosome or Golgi bodies. Fine. Some other cell content are also present. Now after that what we do, we again centrifugate this tube. C means centrifugation. Fine. So C means centrifugation, and now we centrifugate it for 10 to 20 minutes. For 10 to 20 minutes at a speed of almost 8,000 to 10,000 speed. Now at that speed we again centrifugate it. Fine. So now after the centrifugation, what we get? After the centrifugation, what we get? We get the supernatant once again, but within this supernatant there are the active viruses present. Fine. Now in this supernatant, not only active viruses, there are also inactive viruses are also present. Means incomplete viruses or inactive viruses are also present within each supernatant. Fine. So active viruses and inactive viruses and complete or incomplete viruses are present here in this supernatant. So we also get some precipitation like things inside the bottoms. Fine. So heavy particles which are present inside each mixture. So the heavy particles will settle at the bottom, and the light particle, just like the viruses, they will remain on the upper layer of the this solution, and then we take out again each of the viruses into a new tubes. Let's suppose this is a new tube. Fine. Now we again take the viruses within a new tube, and we take these viruses within a new tubes. But there is actually one problem. Now, one problem is actually that we discard this precipitation or pellet form, but we take the supernatant and we shift it into a new tubes. But you know, these new tubes will not only contain the active viruses, there are also some inactive viruses or some incomplete viruses are also present. So after that, we again centrifugate this tube. We again centrifugate for 1 to 3 hours at 1 lakh, sorry, at 10,000 speed. So on that speed for 1 to 3 hour time, we again centrifugate this. Fine. So once we centrifugate this, so what we get? We again get the viruses, but now this time the viruses are now in the precipitation form. Fine. So the viruses are now in a precipitation form. So what we do after that? After that, we again, now to remove this precipitation inside the viruses, as we centrifugate this, now to remove this precipitation, now this is a virus is present but in a precipitation form. If you want to remove this, we put sodium benzoate. Now sodium benzoate, what actually do? Sodium benzoate solution we put. Now sodium benzoate, actually once we put that, so these viruses are washed. So once these viruses are washed, so the precipitation will be eliminated, or the precipitation process will be finished, or the precipitation will be ended. So the precipitation will be ended once we loaded the sodium benzoate. So what we loaded here, we loaded sodium benzoate. So we loaded here sodium benzoate. So what we get? So we get not only the viruses, fine. So we not only get the viruses, mean the sodium benzoate also present. These black dots, let's suppose these present the sodium benzoate which are present, and they are washed out the precipitate, and means they purified the viruses now. But this viruses still contain the sodium benzoate. Now how we remove the sodium benzoate from this solution, and finally the viruses will be in a purified form. So how we will eliminate these means the sodium benzoate. Fine. So once we get this form, and once we get the viruses with the sodium benzoate, so we eliminate it again, we centrifugate it. So we eliminate it with the help of centrifugation once again. So once again we centrifugate it. So once again we centrifugate it. But this time the centrifugation speed may be, so we again centrifugate it at a speed of 5,000. At a 5,000 speed, 10 minutes. Fine. So for 10 minutes we centrifugate it for a 5,000. So we centrifugate it on a 5,000 speed for 10 minutes. Once we centrifugate it, so what we get? Once again we get the once again we get the supernatant, means the virus is in a supernatant form, and the sodium benzoate are now in a precipitation form, or we get the debris, mean the sodium benzoate now in a debris forms. Now these are the debris and these are the viruses in a supernatant form. After that, what we do, we eliminate this sodium benzoate from the tubes, and then we load it here sucrose solution. We eliminate this debris and inside this tubes, then we loaded the sucrose solution. Fine. So we eliminate it, and then we loaded here sucrose. Fine. Actually, what the sucrose do, sucrose create a 3 layers. The sucrose create actually 3 layers. Fine. So we get 3 layers. This is the first layer, this is the second layer, and this is the third layer. We get the 3 layers. At the top of the layer, the sucrose will be 20% present, 20% the sucrose. And at this layer, means at the rest of the 2 layer, the sucrose will be present 50%. Fine. So at the top, sucrose will be present 20%. At the rest of the 2 layers, so sucrose will be present 50%. Fine. Now after that, what we do? So once we get the sucrose, once we put the sucrose, so the viruses which are present in this case, they will be, not only the viruses will get on the upper layer, the other content, cellular content or other debris or the sodium benzoate, if the remaining sodium benzoate are present, so they will be also shifted at the upper layer, and the sucrose solution will also present here. So again, we get 3 things. We get not only 3 things, we get also some other mixture of the solution. We get the sucrose, we get the viruses, complete viruses, incomplete viruses are also present, and we also get some the protein or other debris solutions. Fine. Now why we put the sucrose here? We put the sucrose here for this purpose, that we activate all of the viruses, or we activate all of the viruses once again. That's why we put the sucrose. Although the virus is present in each of the tubes in active form, but still we put the sucrose for this purpose, to activate more viruses. Fine. So that's why we put the sucrose for this purpose. So by loading sucrose inside this, so we get the 3 layers, and the upper layers, there will be our solution. Means viruses, incomplete or complete viruses are present. Fine. So after that, what we do, we again centrifugate this. But now this time we centrifugate this at that speed, means 2,26,000. At that speed we centrifugate the tube once again at a very big centrifugation machine. On a very big centrifugation machine, on that speed we centrifugate the tubes once again for 90 minutes. For 90 minutes we centrifugate it. After this centrifugation, what we get, although it contains the 3 layers, so what we get, we get the 3 layers although. Now at the first layer, there are the protein debris, protein or some other cell content, or there are the debris, simply we can say. Now these are the debris. Fine. At the middle we get the, let's suppose these red one represents the incomplete viruses. Fine. So we get the incomplete viruses. And at the end we get the complete viruses. These green one represent the complete viruses, or loaded viruses, or active viruses. Fine. So now let me write complete loaded viruses. So we get these kind of viruses, and after that what we do, we eliminate these viruses by putting the pipette at this layer, and we suck these viruses with the help of pipette. Once we suck these active viruses with the help of pipette, and then we shift it into a new tube, because we need these viruses. And now inside this new tube, finally active viruses are present, and finally the requirement viruses which we required, these viruses are now present. And so what we get, finally we get the viruses. Now once we get these viruses, actually we need these viruses, and once we get these viruses, then we proceed these viruses under through different tests. Means then we check it under the microscope, electron microscope, under the X-ray diffraction, what kind of virus actually it is. Fine. So that's why we do the whole process to eliminate out the active viruses and to check which kind of active viruses actually that is. Fine. It is really complicated process. Whenever we perform this process inside the lab, so it is very complicated process. Although I present these topics in a very easy way, but still it is very complicated process. Fine. So that's all about the virus purifications. Now let me explain the process once again in Urdu, that what happened. असल में हम ये टॉपिक जो प्रेजेंट कर रहे थे, ये वायरस प्यूरिफिकेशन के हवाले से था कि वायरस प्यूरिफिकेशन क्या चीज़ होता है। वायरस प्यूरिफिकेशन का मतलब यह है कि आपके पास एक ट्यूब है, उसमें आपने पेशेंट का सैंपल डाला है, यानी वो पेशेंट जिस पर वायरस ने अटैक किया हो। अब उस पेशेंट से हमने ब्लड सैंपल या यूरिन सैंपल लेकर हमने इस ट्यूब्स में डाल दिया। अब इस ट्यूब्स में आपके पास ये जो ग्रीन कलर के है, ये वायरस प्रेजेंट करते हैं, जो कि इनकम्प्लीट वायरसेस है। इसके अलावा ये जो रेड कलर के होते हैं, इसमें जो रेड कलर में ड्रॉ किया, ये राइबोसोम्स या दूसरे सेल के कंटेंट्स मौजूद होते हैं। और ये जो ब्लैक कलर का बड़ा सा सेल है, इसमें आपके पास इन्फेक्टेड वायरस जो यहाँ पे प्रेजेंट होते हैं। ये वायरस असल में एक्टिव होते हैं और ये वायरस हमें चाहिए। हम देखना चाहते हैं कि ये असल में कौन से वायरसेस है। तो इस मकसद के लिए हम क्या करते हैं, हम इसमें डिटर्जेंट और बफर डाल देते हैं, जिसकी वजह से ये सेल का जो मेम्ब्रेन होता है वो बर्स्ट हो जाता है, और जितने भी एक्टिव वायरसेस या मेच्योर वायरसेस होते हैं वो बर्स्ट आउट हो जाते हैं। फिर चूंकि हमें ये चीज़ें तो नहीं चाहिए, इसमें जो माइटोकॉन्ड्रिया और दूसरे जो कंटेंट्स होते हैं हमें तो ये चाहिए नहीं, तो इस मकसद के लिए हम सेंट्रिफिकेशन का प्रोसेस करते हैं। सेंट्रिफिकेशन तकरीबन 10,000 स्पीड हम सेंट्रिफिकेशन को लगा देते हैं 1 से 3 घंटे तक, और 1 से 3 घंटे बाद हम इस ट्यूब्स में देख लेते हैं कि हमारे पास एक सुपरनेटेंट बन जाता है। तो सुपरनेटेंट मतलब ये हुआ कि इस सुपरनेटेंट में आपके पास वायरसेस जो होगा वो इनएक्टिव फॉर्म में होगा, और यहाँ पे आपके पास जो ये वाला साइड है, इस साइड पे आपके पास प्लेट वगैरह मौजूद होंगे। इसके बाद फिर क्या होगा, इसके बाद आपके पास ये दूसरा जो ट्यूब्स है, फिर इसमें हम ये सुपरनेटेंट वायरस जो है वो डिस्कार्ड कर लेते हैं। क्योंकि ये आपके पास इनएक्टिव वायरसेस है, हमें इसकी ज़रूरत नहीं होती। इस प्लेट में आपके पास एक्टिव वायरसेस भी होते हैं और कंटेंट वगैरह भी मौजूद है। तो इसको हम क्या करते हैं, अगेन इसको यानी रीसस्पेंड कर लेते हैं, मतलब इसको दोबारा यानी सोल्यूशन फॉर्म में बना देते हैं। जब हमारे पास दोबारा सोल्यूशन फॉर्म में बन गए, तो इसमें अब एक्टिव वायरसेस भी है और दूसरे सेल कंटेंट भी है। ना सिर्फ एक्टिव वायरसेस बल्कि इनएक्टिव वायरसेस भी मौजूद होते हैं। फाइन। तो फिर हम क्या कर लेते हैं, फिर दोबारा हम इसको सेंट्रिफ्यूगेट कर देते हैं 8,000 से 10,000 स्पीड तक, 10 से 20 मिनट तक। फिर हम देख लेते हैं हमारे पास दोबारा फिर प्रेसिपिटेट बन जाता है, और ये वायरसेस जो हमारे पास एक्टिव वायरसेस होता है, फिर हम इसको क्या करते हैं, एक दूसरे नए ट्यूब्स में शिफ्ट कर लेते हैं। अब जब हम इसको एक नए ट्यूब में शिफ्ट कर लेते हैं, तो ये वायरसेस तो इसमें ना सिर्फ एक्टिव वायरसेस होते हैं बल्कि इनएक्टिव वायरसेस मौजूद होते हैं। अब हम इसको दोबारा सेंट्रिफ्यूगेट करते हैं 1 से 3 घंटे के लिए 10,000 स्पीड पर। तो फिर हम प्रेसिपिटेशन हासिल करते हैं। हमें तो प्रेसिपिटेशन की ज़रूरत पड़ती ही नहीं है। तो इस प्रेसिपिटेशन को हटाने के लिए हम क्या करते हैं, हम सोडियम बेंजोएट से इस वायरस को वॉश कर लेते हैं। जब सोडियम बेंजोएट को हम इस वायरस को वॉश कर लेते हैं, तो दोबारा हम वायरसेस को एक्टिव फॉर्म में या वायरस को फिर दोबारा सस्पेंडेड फॉर्म में हम हासिल कर लेते हैं। अब ये वायरस सस्पेंडेड फॉर्म में है, एक्टिव फॉर्म में है, और इसमें सोडियम बेंजोएट भी है। चूँकि हमें सोडियम बेंजोएट चाहिए नहीं, तो दोबारा इसको सेंट्रिफ्यूगेट करते हैं 10 मिनट के लिए 5,000 स्पीड पर। तो ये आपके पास इसमें सोडियम बेंजोएट एक डिब्रीड्स बना लेते हैं, आई मीन डिब्रीज हमारे पास बन जाते हैं या प्रेसिपिटेशन नीचे बन जाते हैं। हम इसको एलिमिनेट कर लेते हैं, हटा देते हैं। और ये जो हमारे पास जो वायरसेस होते हैं, इसमें एक्टिव और इनएक्टिव वायरसेस भी होते हैं। इस मकसद के लिए अगर हम चाहते हैं कि इस इनएक्टिव वायरसेस को भी हम एक्टिव कर लेते हैं, तो हम इस मकसद के लिए क्या करते हैं, हम इसमें सक्रोज डाल देते हैं। सक्रोज जब हम डाल देते हैं वो इसके लिए एक न्यूट्रिएंट के तौर पर हम कह सकते हैं कि काम करता है। तो सक्रोज यहाँ पर 3 लेयर बनाते हैं, जैसे कि 1 2 3, जैसे कि मैंने आपके यहाँ पर ये लेयर जो बनाए हैं, इसमें 20% ऊपर होता है और 50% नीचे होता है। लेकिन जो 20% होते हैं वहाँ पर वायरसेस और दूसरे सेल कंटेंट्स चले जाते हैं ऊपर से हिस्से पर। अब इस ऊपर वाली लेयर पे आपके पास ना सिर्फ वायरसेस है बल्कि इसमें सक्रोज के सोल्यूशंस भी है और दूसरे जो सेल कंटेंट, दूसरे डिब्रीज वगैरह भी यहाँ पे मौजूद होता है। फिर हम इसको तकरीबन 2,24,000 स्पीड पर हम इसको चलाते हैं एक बहुत बड़े सेंट्रीफ्यूगेशन मशीन में। सेंट्रीफ्यूगेशन तकरीबन 90 मिनट तक हम इसको करते हैं। फिर इसके बाद हम इस ट्यूब में क्या देखते हैं, एक 3 लेयर्स देखते हैं। इसके अपर लेयर्स में आपके पास प्रोटीन, दूसरे डिब्रीड्स वगैरह मौजूद होते हैं। इस मिड लेयर में आपके पास इनकम्प्लीट वायरसेस मौजूद होते हैं और इसके जो आखिरी हिस्सा है, जो आखिरी लेयर होता है, उसमें कंप्लीट वायरसेस, लोडेड वायरसेस, इन्फेक्टिव वायरसेस मौजूद होते हैं, जो कि असल में हमें चाहिए यही वायरसेस। तो हम पिपेट को अंदर कर लेते हैं और हम ये वायरसेस लेके नए ट्यूब्स में शिफ्ट कर लेते हैं। अब चूँकि हमारे पास एक्टिव वायरसेस आ गए हैं, हमें इसी वायरसेस की ज़रूरत है और फिर हम इसको इलेक्ट्रॉन माइक्रोस्कोप, एक्सरे डिफ्रैक्शन में देख देते हैं कि असल में ये कौन से वायरसेस हैं। सही। तो नाउ दैट्स ऑल अबाउट द वायरसेस प्यूरिफिकेशन। आई होप दिस वीडियो हेल्पफुल इफ़ यू अंडरस्टैंड. So best of luck. If you have any kind of question, so write a question into a comment. I will try my best to give you answer as soon as possible. So please like the video, share the video, and don't forget to subscribe the channel. Subscribe the channel to get more interesting video lecture like that. Thank you so much.